And in this series, we begin with Dr. Anna Barron from Nanyang Technological University and EMBO Global Investigator. Today, she will talk to us about the journey through the brain metabolism and discovery.
Good afternoon, everyone. I hope everyone's feeling rested after their lunch and the exciting sessions this morning. So I'd just like to start by introducing, it's part of a EMBO Global Lecture Series talk. So this is funded by EMBO, and I think you've heard a lot about the EMBO programs this morning from Gerland. So I hope you're excited about this. So I'm actually a global EMBO investigator. And that's actually how I got invited to this. So I met Neha through the EMBO, the Global Investigator Program, and that's how we know each other. So EMBO, just to give you a little bit of background about EMBO, I think you probably already heard a lot, but we'll just touch on it again. So EMBO, it's the European Molecular Biological Organization, and there's over 2,000 members, and this includes not just the European states, but also here, India, and where I'm based as well, Singapore. So everybody here who's based in India is also eligible to apply for these programs. There's a lot of different postdoctoral fellowships, so people from your lab can apply for this, or yourself if you're still a postdoc. There's scientific exchange grants, and this is available for people at all levels of their career. There's also so funding, for example, like we have here for lecture workshops and to facilitate networking activities. And then also funding for early investigators like what I myself got through the Global Investigator Program. And also just a plug for their avenues for publishing your work as well. So I myself have submitted work here, and I can attest to the fact that the editorial process is very rapid and fair and efficient. So this is a great place that's very well internationally recognised to publish your work as well. Okay, so to tell you a little bit, I'm here to tell you a little bit about myself and what my journey has been in neuroscience. So I'm a neuroscientist. I'm originally from Australia. And so today I'm not going to be talking too much about the science, so apologies to some of the people who have come and spoken to me and said they're really excited to hear about some of the science we're doing, but we're not going to focus on that too much. Instead, I'm going to be telling you more about what my journey has been like, how I ended up here standing in front of you today. So I'm originally from a regional Western Australia. So this is a picture of what you see when you look outside the backyard of my hometown, of my childhood home. So I grew up amongst horses and chickens and these kinds of things. And so when I graduated from school, I decided to go to university because I was really driven to get out of that very small town. And I decided to study neuroscience. So I've been a neuroscientist right from the start. And as perhaps with many optimistic young people, I didn't really put a lot of thought into what my career prospects were. I just thought, wow, this sounds like an amazing frontier. This sounds like something that, you know, is really exciting to learn about. And so this is what I went and studied. And it was correct. It is a really, and still is a really exciting frontier. And it's sustained me since I started this in 1999 to continue working on this. And so I think something that might be interesting for you to know is that when I was studying as an undergraduate, I now teach from the same textbook, obviously an updated edition. But the textbook is actually about double the size. So this reflects the amount of new knowledge since I was an undergraduate that has been generated in the neuroscience field. So it's been quite a rapidly progressing field. So after I finished my PhD, you'll probably notice there's a little bit of a gap here. And so I actually worked as a technician for a couple of years. And that was a really transformative time for me, actually. I was taken in by a mentor who was really nurturing. She treated me like a PhD student. She gave me my own project to work on. And I learned a lot of the soft skills that really made my PhD go very efficiently. And beyond that, also, it allowed me to really identify what it is I wanted to work on before I embarked on a PhD. So it meant once I got to the PhD stage, I was really driven. I'd selected my topic based on something I was really passionate about. And so to give you a little bit of insight into that, this is actually me when I graduated from my PhD with my very dear grandmother. So I lived with her when I first started going to university, and she was actually a carer for my grandfather who had Alzheimer's disease. So I got to see quite firsthand what the disease process is. So this was something that I felt would be meaningful to devote my career to. Also, sorry, I forgot to mention, I had the opportunity in my PhD to go overseas. I went to NYU, New York University, and spent a short period of time there. And that also had a really big influence on me because the way they did science there was completely different to the way we did science in Australia. I think somebody earlier today we heard about how you can order something, it'll come the next day, these kind of things. Australia, we suffer the same problems. You wait 6 weeks for anything you order to come. You don't have the same resources as what are available in the US. So often if we needed new equipment, we would go to the workshop, work with the people in the workshop to draw up a plan of what we'd like them to build, and they would manufacture or fabricate something for us to fit our solutions rather than purchasing pre-constructed materials. So after I went to NYU and I finished my PhD, I wanted more of this fast science that I'd experienced in the US. So I looked at a few different postdoc options, and I ended up going to University of Southern California in LA and joining Christian Pike's group there. And this was a time so my research focus had been Alzheimer's disease, looking at how hormone replacement therapy could potentially be used as a treatment in Alzheimer's. And towards the end of my PhD, there was a large clinical trial known as the Women's Health Initiative, and actually it was a massive failure. And this was despite all of the really good basic science, preclinical evidence that this trial should work. And so what my postdoc was really focused on was what went wrong. What went wrong between the translation from the basic science into the clinical trial, that this huge amount of data that says this should be protective, it actually had the opposite effect when it went into the clinic. So that was what I was working on in my postdoc. And you might notice these little disasters that happened here. I started to feel a bit like every time I moved to a new place, I was bringing a disaster with me. So just after I moved to USC, or actually just before the global financial crisis hit, and so this actually really affected the landscape. I'd gone there to work in a lab that was really well funded and that had a lot of resources and these kind of things. And actually I walked into an environment where 2 people lost their jobs. There was 5 postdocs when I started. When I left, I was the last postdoc standing. And the lab was like running out of money. The pay line for NIH was below 5%. At this stage, I actually got some independent funding while I was here at USC as well. And this gave me, so I had a lot of opportunity to write grants together, frantically writing grants together with Christian while we were trying to get more money to keep the lab going. And so when I left, it was just 1 student that was left in the lab. And I instead started focusing on, I wrote a proposal to go to Japan. And so the reason what motivated me to go to Japan was actually, I was really interested in a particular technique that was only available in this lab in Japan, based in Japan. And so I went there to learn it, and I wrote a proposal to the JSPS Foundation, which is like the National Science Foundation for Japan. And so I had a 2-year postdoctoral fellowship there. And shortly after I arrived, there was the Tohoku earthquake a couple of months after I arrived. And then shortly after that, there was also the Fukushima nuclear disaster. And so this was a little bit of a crazy time. So I think everybody's familiar with what happened in the COVID pandemic and how surreal all of that was. Well, I think this time in Tokyo was also quite similar. So actually the day before the meltdown of the Fukushima nuclear reactor, my PI sat me down and he said, "Look, there's no reason to panic. This is not going to melt down. Everybody's staying here in the lab. We're going to be coming to work every day. We just continue on. There's nothing to be worried about." He had printouts of the nuclear reactor and why it was no problem. And then the next day I came to work and he said, "There's nobody left, everybody ran away." And by that stage, all of the flights had shut, actually. And so the only people who could be evacuated were people that were being evacuated by the embassy on embassy flights. And so I was stranded in Tokyo under these conditions. And so one of the interesting things was that we actually worked in a radioisotope facility. So we could test ourselves each day and our food and things like this to check for if we had been affected by any contamination. And you might think, oh, Tokyo, it's quite far, right? Well, that's what I kept telling my family when they were calling me up every day saying, like, why are you still there? But it's really dependent on the weather as to how far the radiation goes. And so every day we were watching the weather. And after the meltdown happened, we were advised that you needed to stockpile water in case the weather transported the radiation to the reservoirs and you wouldn't be able to access clean drinking water anymore. So, for example, we filled our bathtub with water that was, in an emergency situation, would be all that we had to use. So yeah, there was, I would say, a little hiatus in research and some disruptions that occurred during that time. But there was lots of really good things that happened as well. So this is one of our lab parties. I'm not sure if anyone's familiar with something called suikawari. It's where you have a watermelon and a big stick and the person's blindfolded and you take turns at trying to whack the watermelon with the stick and crack it open and then you eat it. Okay, so towards the end of this postdoctoral period in 2012, I started applying for independent positions in Japan. So I wanted to stay in Japan. I'd had a really productive experience there. I'd really enjoyed the landscape of how research happened. It was very culturally different to what I was used to, but it posed a challenge that I was enjoying, and I think the funding is much better than Australia, and the quality of science I was really enjoying as well. So I started looking for faculty positions there, and I made many, many, many applications. I'm not sure how many, and I didn't get a single interview, except I think for one, and I really felt like I was a zoo exhibit when I went. They were very curious as to what this Australian woman was doing wanting a faculty position in Tokyo. So after I finished this postdoctoral fellowship, I actually, through one of my applications, met these researchers at the National Institute of Radiological Sciences. And it turned out that we had overlapping research interests. And so this kind of changed the trajectory of my research a little bit. So we were both interested in some mitochondrial aspects in the context of neurodegenerative disease. And they were developing some transgenic models on some of the molecules that I was really interested in. And I had some expertise that could help them. So I joined this group in 2013. And I started out as a postdoc, and then I got promoted to a tenure-track researcher position. So this is not a university, but like a national institute. And so the tenure-track process there, culturally, it was a little bit different to what I was used to. So I was considered very young to be given a tenure-track position, whereas I think internationally I would have considered myself quite old. And there's kind of a queue that you get in to get your tenure. So people that have been there for longer than you, it's not necessarily a merit-based system. It's everybody who has a position there. If they keep their position, then you're considered to have sufficient merit to get tenure, and then you wait your turn in the queue. So I would say that my time here was really, really productive. And I really enjoyed working with this group. I was given by my mentor, the first thing I noticed when I joined this group was that everybody loved him. When we would go to lunch, if he came to lunch with us, everybody was just so excited to talk to him and, to want to discuss science with him. And I really saw that the way he interacted with the group affected the entire group dynamics. And, I knew there was a lot of politics that occurred in the department, but he really shielded us from all of the politics. So I think I really learnt a lot about mentorship from the experience of working in this group. And then, so in 2015, I kept applying for independent positions unsuccessfully. And then in 2015, I had a side project. So my son was born. And I took some maternity leave that went on into 2016. And around this time, I also applied to this program in Singapore. So this is at NTU, and they recruit across all disciplines internationally. So it's a program aimed at recruiting people internationally to Singapore. And I got an interview. My son was 6 weeks old when I went to the interview. And to be honest, I just wanted to communicate my science to people, and I had never been to Singapore before. I didn't really know anything about Singapore except it's, you know, people like it for shopping. That's not me, so couldn't imagine myself living there. And I went to the interview, and I was really, really impressed with the quality of the science, the interactions with people, and I ended up getting the position. So in 2017, I waited. Oops. Hang on, we're skipping ahead. So I waited, I was offered the position in 2016, but I delayed starting for a year because I wanted to finish up my work and also complete my maternity leave and these types of things. So I delayed starting until 2017. And then in 2017, I moved to Singapore to start my lab at NTU, Nanyang Technological University. So this is in the medical school, the Lee Kong Chan School of Medicine. This is a relatively new medical school. It's the newest medical school in Singapore. So it's about, I think we're coming up to 15 years old now. And so when I joined, it was around 5 years old, I think. And so this is the space that my lab would eventually be in, and you'll see why I say eventually in a minute. This is we have a dual campus. So this is our the main campus. It's out in the jungle, essentially, and I think Gerlind has had the pleasure of being there. And so the campus which I'm based on, we're attached to one of the hospitals so that we can collaborate effectively with the clinicians, and also for teaching purposes as well, because we're a teaching school. So initially, when I first moved there, actually, within the first 2 years, we moved lab 3 times. So initially, we had a footprint here in Singapore. Singapore is very small. I think it's about 50 kilometres across. So this probably sounds quite trivial. But initially, we were here, then we moved here, and then eventually we moved here, which is the final place that we've set up lab. So this was, you know, I mean, it was quite disruptive to the group. There was, you know, there was lots of other teething problems. Sometimes things that you're promised don't materialise or eventuate to be what you thought they were going to be. And I think the take-home point on that is that everybody has problems. And, you know, life is unfair, and that's not unique to science. That's true of whatever, you know, field you will work in. And don't dwell on it too much. Just, you know, get the job done and then move on to the fun stuff of doing the science. So after I think I'll backtrack a little bit to how I went about building the team. So one of the first things that I did was when I negotiated my startup, because I knew I'd be delaying starting the lab, I actually asked if I could have a bridging fund and a small footprint of bench space to begin setting up my lab before I was on the ground in Singapore. And that included several trips for me to come in person and, you know, meet the lab member that I would be recruiting and help, you know, facilitate things moving on the ground. And so this was something that was actually really beneficial. I was using transgenic mouse models. So they take a really long time to establish. I was importing them from Japan, so there was a lot of paperwork to be done. And so I'd hired someone that was on the ground on the other side that could help coordinate all of these things, so that when I finally did reach Singapore to start the lab in person, a lot of things were already underway. I'd got a lot of approvals that I needed, for example, my ICOOK approvals, IRB approvals. A lot of these types of things were already in the process. So I think that if you have the opportunity to do that, you don't need to ask for a lot, but it can make quite a big difference if you can start to get these things running, especially if you're on a tenure clock. And so one of the first pieces of advice that anybody gave me, which I didn't like very much, was that you will be your best postdoc for a long time. As a new, unknown PI, you're not going to be able to recruit high-flying postdocs and high-flying top talents. You know, you're not going to be getting students from Harvard, this kind of thing. And so really you have to be at the bench and doing a lot of the work physically yourself. And I think this is really true. And I think that people who are successful tend to take this approach. So the other thing, I guess, along this line is that because you're not going to be, taking Harvard graduates, you really need to hire for the potential of the people. You need to find the diamonds in the rough. So how do you do this? Well, my strategy was that, I think based on my own experience, that I really value people who have non-linear trajectories. So people who, for example, may have gone out and worked in a different field before they have decided, hang on a minute, this is not what I want to be doing. I really want to do science, and they come back and they study their PhD. These people tend to be really committed. They know what they want to do. They've got a lot of drive. And so I think you can't make up for that, having that grit. Also, a strong sense of purpose, and also that you have aligned values. And you'll hear me say this quite a number of times, but I don't think I can stress enough how important it is that you have aligned values. So I think a lot of us spend a lot of time thinking about our research questions, but not enough time thinking about what are the values that are driving our research questions and embodying in our science. And then also, I really wanted to make sure that I diversified the lab. So the temptation, I think, is, you interview someone, you really like them, and it's because they think like you, and you can understand them, where they're coming from, their training, what they're thinking. But there's no point in hiring a carbon copy of yourself. You need to hire someone who's going to add value and diversify the perspectives, the experience, the skill set that you get brought to your lab. I think another really important thing was to build the team together. So once you start recruiting people into your lab, you need to involve them in this decision-making process. That's how I've always approached it. I always get any new potential recruits to talk to the lab members. And then I don't give them necessarily the decision making, but I ask them what they think about the new person. Could they work together? Is there anything that rings alarm bells for them? These kind of things. Because you'll find that people will interact very differently with you than they will with other team members. So sometimes you don't really see all the facets of a person. Obviously when somebody's in an interview, it might be quite different to how they behave at the bench. And then, so I think the last thing on this is some of the lessons that I learned, perhaps the hard way, is that you need to fix your hiring mistakes quickly. And so I definitely made mistakes in hiring. It took me a number of years to sort of equilibrate my lab correctly. But the problem is that your whole lab suffers as a consequence of these mistakes. And so, I think it is really hard to do, but you have to figure out a way to manage people out to protect the group. And it's better to have no one than to have the wrong person, I think. So once you have your team, how do you grow your team? Well, I think this is really important because often we think science is about our projects, it's about a question, but it's not only about that. Actually, the people are really important too. And so we talk a lot about being curious in science, but I think we also need to be curious about our people, the people that are in our labs. So, what do they want to achieve? Are you aligned in what you want to achieve? This should be a symbiotic relationship where there's alignment between what they want to achieve and what you need to achieve. What do they need to succeed, and can you provide it? And so this requires some tough introspection and some honesty with yourself about where your strengths and weaknesses are. What are their expectations, and do they match your expectations? If there's an expectation misalignment, anyone who's a neuroscientist will know this leads to disappointment. So you need to know what you can and what you can't do. So I touched on that one in the past point. And it might take you a little while to really figure out what these points are, but they're really important to know what your limitations are. And when you can't provide the support your people need, you need to be really proactive in finding someone who can provide that support. Connect your people with people that can expand their world. And so I think one of the most important things, we talk a lot about being a good mentor, but from my perspective, it's not just about being a good mentor. What you really need is an advocate. And this is true not just for our people in your team, but for yourself as well. So the difference, you might be thinking, what's the difference between a mentor and an advocate? So a mentor is someone who guides you, whereas an advocate is someone who will advance you. And I think if you think about your experience and interaction with people, you'll probably be able to identify the difference in some of your previous mentoring relationships. So you need to create opportunity, visibility, and protection for the people in your team. And you're also going to need to seek that for yourself. And you'll find as you rise up higher, it's harder to find advocates, actually. Although you'll have an abundance of mentors, perhaps a dearth of advocates. Okay, so beyond growing your team, you also need to lead your team. So what I think is really important in leading your team is, the number one thing I think is leading your team by example. So this is, I mentioned this before, that you really need to carefully define not just what your scientific goals are, but what your values are as well. And so these are really intricately linked, right? And you only need to understand this if you think about, what does the term good science mean? Right? Good science is a very value-laden term, and it embodies what your values are. And so this also will affect what choices you'll make, and it's important that you, as a leader, you have a clear vision for both your values and also your scientific goals. I find it really helpful to actually crystallize this, write it down, discuss it with the team, avoids any mismatch of expectations. And then I think the most important thing is that you really need to, I found I needed to demonstrate this in practice consistently to the team. And so this is not just in my interactions with them, but my interactions with everybody. And I think admin is something that has come up quite a bit. And I think this is one example of where it's really important to demonstrate in practice what your values are. So sometimes there can be really frustrating situations, but at the core, what my value is that the admin support is part of the research team. They're critical for us achieving our goals. So my approach to it is that I try to bring them into the excitement of, we're in this together, they're helping me, we're working together to achieve a goal that I think is really important. The other thing I think is really important is creating a positive culture. So my lab, my office actually looks out onto the lab. There's a big fishbowl window, and anytime somebody has, an experiment, they're getting the result, they come and knock on my window because they know that I'm sitting there waiting to see what their results are. And I'm going to be really excited to, come to the microscope, come to the bench and see what the outcome of their experiment is. And if it's a failure, I'm going to share the disappointment with them, right? So I think that's quite important. And then developing really strong tools for communication. And so just to throw a cat among the pigeons, I know everybody really likes lab meetings. Well, I hate lab meetings. And I sort of developed this in Japan. I have the feeling often it becomes a platform for people to hear their own voices. And so I prefer to keep it as individual or small team meetings. But anyway, I think however you do it, you need to work out really clear processes for communication. And you'll find that, people will communicate differently. Some people will not. I'm a verbal communicator. I found some students, they just forget 2 seconds later what I've said, or maybe they're not understanding. I'm Australian. The way I communicate is quite different to many of the students in my lab, and so it can lead to misunderstandings. So I think doing a reality check with people about, are we on the same page? Have you? If you repeat back to me or email me what you think I just told you, we can do a reality check of, are we all on the same page and moving in the same direction? So I'm not sure what obviously a lot of what I'm talking about might be quite specific to Singapore. I'm not sure what the lay of the land here is in India, but I'll just talk a little bit about tenure. This was like a really, a defining feature of my first 7 years when I was at NTU. So some of the key points that I learnt early on, which I was fortunate enough to learn early on, is that you need to really think strategically about what your niche is. You need to consider how you fit into the institution. And so these things need to play into how you develop your research program and what questions you tackle. It's really important to learn to say no. So up until this point, I'd spent my whole career learning how to say yes to every opportunity that passed my way. And then once I became faculty, I had to start learning how to say no because I was overwhelmed with opportunities and quite time poor now. The other thing I found really important is that you need to find your support network. So you need to find your crew, other early faculty members that share your pain in the tenure process and all the challenges, they understand the challenges you face. And also find your advocates, not just your mentors. And what I found really helped me, and unfortunately this sort of came as a result of one of my really good friends got denied tenure in my first year that I was at NTU, and it completely shook my world because she's an amazing scientist. And it made me realize that, this is real, this is something that can happen. Some of it is, luck of the draw, and it's not necessarily a very transparent process of how these decisions are made. And so, first of all, focus on the science. Don't worry about thinking about the tenure too much. But what I found really helpful was to just think of this as a fixed term position. Okay, I had a 7-year tenure clock. I have 7 years to do something really good. When I leave, am I going to be satisfied with what I've achieved? And then if I get anything more than that, then that's, icing on the cake. But it takes away this stress of, if I just think of it as, it's another fellowship position that I have for 7 years. And I found that really useful for not worrying myself about the tenure process. And then I think the last thing to say is, I've had many friends now that have gone through this tenure denial process. It's not the end of the world. All of those friends have very fruitful careers still. I think often we feel like, a tenure denial is the end of the world, but it's not actually. I don't really have a lot to say on securing funding. I don't know much about the landscape here. But from my experience, initially when I moved to Singapore, I found it easier to get funding internationally than I did within Singapore. And this was essentially because I didn't really understand the landscape and what the funding bodies were looking for. So it's really important to understand that. And then, you need to think about it strategically as well, of what you're applying for. I think I mentioned yesterday, I had a grant that I put in that I thought, I had really good preliminary data, and then a mentor said to me, is this really what you want to be known for? Or is it something that you can get funded easily? And the answer was it was something I could get funded easily. But you get the grant and then you've got to do the work, and then your time is carved up with this. I'm going to skip over this because I see I'm running out of time. Oh, maybe I can take some of the 10-minute discussion. No? Okay. Okay, so if you are at a university that also involves teaching, so we have about 50-50 research and teaching load, and then, I don't know, it doesn't add up to 1, it actually adds up to 1.2. I've never figured out how that works. Then we have a 0.2 teaching load, 0.2 service load. So when it came to teaching, I started getting experience in this really early. I was not trained formally in teaching, and so it was probably the thing that I found most intimidating. So I started, getting involved in teaching right from my PhD, and anywhere I had the opportunity, I got involved in teaching coursework. And then once I did start my faculty position, what I found was learning from my peers. So I co-taught with someone who was quite senior, and they're an amazing teacher, and I learnt so much from just observing how they go about teaching, how they manage students, and that's probably where I learnt the most. I think also, don't be afraid to take professional development training. So I did, formal courses on teaching as well, particularly because my institute, we have a team-based learning approach exclusively, and that was completely new to me. My experience of being a student had always been didactic, that, there's a lecturer, and instead this was very intimidating because it was a very interactive and open discussion with the students, and so it was a very new experience for me. So, I think if you have resources you can access for training, then definitely take them. And then the last thing I'll say on this is that teaching is, how long is a piece of string? You can spend weeks and weeks developing the perfect lecture, the perfect course, but you need to really limit the time you devote to this and treat it not as a final product, but as an ongoing process. So every year you improve it. You give yourself a set amount of time. This is how much I'm going to devote to it. Next year, I'll make it better. And I think that's really important because my first year, I got quite bogged down with wanting to make the perfect lectures, and it can take up a lot of your time. Okay, so the last comment that I want to make is that sometimes where you are doing your research or starting your lab, there may not be all the infrastructure that you want and need. In fact, there definitely won't be. I don't know, no one works in utopia. This is your opportunity to contribute to building infrastructure, changing the landscape and building capacity. And so for me, I had the opportunity to contribute to establishing the Singapore Brain Bank. So this is a nationwide initiative in Singapore. So Singapore has a really genetically interesting population because there's people of different racial descents of Chinese, Indian, and Malay as well. And so that there's no other brain bank in the world that captures these populations living in one particular city. So this initiative was established in 2018. We now have almost 1,000 donors. We have 15 brains in the collection. I know that sounds like not very many, but actually when we started this, we were told it was impossible because culturally Singaporeans would not be willing to donate their brains, but they're wrong. We now have proof in principle that this is possible. We have released our first tissue to researchers as well, so we have, I think, we're at 6 ongoing projects at the moment. And so these are just used as for pilot studies. I think we have around 5 Alzheimer cases and then 5 control cases and then a range of other neurodegenerative and neuropsychiatric diseases. So I would encourage you to really think strategically also about, if your tenure, you don't get tenure, what will be your legacy? What do you want to have contributed and changed the landscape in your institute and in your community when you leave? Okay, so I'm kind of out of time, but I'll just tell you a little bit about my science. So my research interests, I'm interested in brain aging and particularly Alzheimer's disease. And so I'm really interested in how metabolism affects this, and particularly the bioenergetic organelle of the cell, the mitochondria. And so the brain's really interesting because it's really highly energetic. So although it's only 2% of your body weight, it actually uses around 20% of all of your energy that you require. And this is when you're at a resting state, so not when you're thinking. And so it's really susceptible to energetic disruption and mitochondrial dysfunction. And so these are the questions that my lab is interested in addressing. How is this mitochondria disrupted in brain aging and disease? How can we measure this? So this is looking at biomarkers. And how can we treat it? So looking at potential therapeutics. So our approach to answering these questions is that we're looking at developing mechanistic understanding. And so we have a few different projects that we're working on towards this. This was born out of necessity that we needed to work on tool development because a lot of the tools for studying this problem are very limited. And then we're also working to develop a preclinical foundation for the development of both diagnostics and therapeutics. And so our approach to this is multi-pronged, and don't be intimidated by this. This is not what it looked like when my lab started. It probably looked like cell culture models and mouse models. Now we also do zebrafish as well. But so we take a multimodal approach looking across levels of molecular pathways, cell processes, so the cell biology and functional outcomes through to organ systems. And we use multi-omics approaches, functional assays, and then in vivo models as well. We use patient samples, both post-mortem brain tissue, and then we also work with clinicians for peripherally derived blood cells, so looking at the immune populations, and we use a range of transgenic animal models, both mouse and zebrafish. And so I was going to give you a case study of some of the work from our lab that is looking at a particular mitochondrial molecular marker that's often used as a marker of inflammation. And so this is quite a translatable tool because it can be used in PET imaging, so you can visualize this in the living brain in both animals and humans. And so our question really was, what is the molecular function of TSBO? Because this is unknown. It's been identified both as a therapeutic and a diagnostic, but nobody really knows what is this mitochondrial protein doing that it regulates the immune function of these cells. And so this was kind of a funny story because we found that, so we took the knockout approach, we knocked this mitochondrial protein out and it had all of these metabolic effects that resulted in all these functional changes that basically exacerbated Alzheimer's-like symptoms. and made the microglia, the immune cells of the brain, quite dysfunctional and unable to clear away waste in the brain of the Alzheimer's. And so what we found was there was a change in this glycolytic enzyme, in the activity of it. But what was really weird was there was no change in the levels of the enzyme. So both transcriptionally and at the protein levels, we thought for sure when we look at protein levels, there'll be a difference in the amount of this enzyme there. And my student came back and she said, no, it's exactly the same amount. And so I went and opened up all her files and looked at the images, and I was like, but it looks different by eye. And she said, yeah, I noticed that. It seems like it's stuck all over the mitochondria. And I was like, well, that sounds kind of important. And so she went away and quantified it. And indeed, what happened is that there's a change in the localization within the cell. Although there's no change in the total amount of this enzyme, where it's located within the cell changes. And so we did a lot of work, and we could show that this basically acts like a molecular switch for controlling the metabolism of these immune cells in the brain. And this also then controls the function of the cells, the immune function. And so this is really important in Alzheimer's disease for a really important protective mechanism, which is clearance of these toxic aggregates. So we then leveraged this to develop an optogenetic tool. So this is basically a light-activated molecular switch that we can control glycolysis or the metabolic activity of our cells. So it's genetically encoded. We've made transgenic fish with this. We're now in the process of making mouse models expressing this, where we can basically shine light in the brain and non-invasively control the metabolism of these immune cells.
Anna, sorry, can you please conclude?
Yes, I will finish. Okay, so the last thing I want to tell you is who are we now? So this is my lab group at the moment. I have a couple of postdocs. I keep my lab group deliberately quite small. I have 3 PhD students and some undergraduates that usually they stay for a 1-year attachment project, and a technician. And I would say all of these people, they're very talented. They've applied for a lot of different awards, conference awards. They've won a lot of different awards. For example, Kayon at the moment, she's actually seconded to Cambridge University, and then she'll be coming back next year to complete her postdoctoral fellowship. So on that note, I will open up for any questions.
Can I take 1 or 2 quick questions?
Hi, Anna, really cool work. My name is Priyanka. It was really interesting to see that TSPO is one of the genes that you're studying in Alzheimer's. I'm trying to model PTSD, and more recently TSPO has been identified as one of the, well, not risk, because PTSD doesn't really have risk, it's more like the illness state gene. And parallelly with our work with autism, we are noticing a lot more of mitochondrial function coming up there as well. So, I mean, my question is more in regards of, do you think that in a neurodevelopmental versus a neurodegenerative state, is the mitochondrial function going into sort of a suboptimal function later on, or could there be more developmental? Could these genes be switching early on in development that is ending up in a degenerative state later in the age or something?
Yeah, so I think that's a really interesting question, and actually that's with our optogenetic tools, that's kind of where we're going now. So there's a lot of parallels in the immune states we see in neurodegenerative disease with the immune states in the context of neurodevelopment, in the fate specification of, for example, microglia. And so we're really interested in how metabolic changes in the context of development might play a role in the fate programming of the innate immune cells. And so that's something we're investigating with our optogenetic tools. And so we've actually done some of these experiments, and I've been trying to move away from TSPO actually, and so to my surprise / horror, I don't know what, the top differentially expressed gene, guess what it was? TSPO. Yeah, after we inhibit glycolysis, it was TSPO. Yeah.
Okay. I have follow-up questions, but
Sure.
later.
Ma'am, I have one doubt. Since aging in the brain, you cover the whole area of the brain, But so you have looked only for the mitochondrial disruption in microglia. Are you looking for it in other brain cells and other immune cells in the brain too, like astrocytes and neuron cells?
So I think, calling neuroscience neuroscience was a bit of a misnomer and has misdirected a lot of research. But yes, we are looking at neurons. So we're looking at neurons and microglia. We don't do any work on astrocytes, actually. I think that field of looking at the role of metabolism in astrocytes is quite populated already. I didn't show any of the work we do on the neuronal aspects in the context of neurodegeneration. But I think microglia are really interesting because they have very flexible metabolism. So compared to neurons, they have very inflexible metabolism. They're very susceptible to mitochondrial damage as a consequence. But microglia, they actually have very flexible metabolism, and this is very, very closely linked with their function. So we're interested in how we can target this to repair their function. I think there's a lot of work already looking at neuronal dysfunction in neurodegenerative disease and how we can potentially target that. Yeah.
Thank you.
Let's thank Anna for the excellent talk. Thank you, Anna.